All right, well, first of all, just thanks for taking time out of your guys' day to come hear what I've been doing for the past 5 months or 6 months or however long I've been here. I appreciate you guys taking the time to listen. So I've been working mostly on seagrass restoration in the Caloosahatchee, Rick said. So I'll just give a little bit of a brief history, background information, and then take you through kind of our methods and the results that we've gotten so far on the project. So why is seagrass important? A lot of different reasons. It's really important as breeding and nursery ground for fish. So that's obviously really important for sustaining fish populations and fisheries. It's also a huge carbon sink. It's been estimated that it accounts for approximately 50% of the carbon burial in the ocean, so that's pretty huge. And it's also food for keystone species such as manatees and sea turtles. And it also provides coastal protection and erosion control, which is obviously very important. And it does that through the dissipation and attenuation of wave energy. So it's basically just reducing the wave energy before it actually hits the coast. And it's also a big improver of water quality. And it does that by taking up nutrients and pollutants from the water. But seagrass, unfortunately, is under a lot of pressure for a lot of different reasons. In a very comprehensive literature review, they found that 29% of seagrass beds worldwide are either lost or degraded. So we're losing these habitats very quickly, and a large percentage of it. And they're under a lot of pressure from mostly anthropogenic reasons. A big one is through nutrient and particle pollution, so that's through added runoff and it can development and things like that are adding excess particles to our aquatic systems. And it affects the seagrasses by it reduces the amount of light that can reach the seagrasses through direct shading from the particles, or it can be promoting the growth of phytoplankton and epiphytes, which are the little organisms that grow right on the leaves of the seagrass. So all things that are reducing the amount of light that the seagrass can access. And other ways that we're impacting seabeds is through the seagrass beds are through physical modifications, so coastal development, anything where you're changing the topography of the bottom. And also through overexploitation. Seagrass has been used around the world for food, for fertilizer, and just as raw materials. And also obviously climate change, as with everything. Global warming and sea level rise is pushing the seagrass to kind of the limits of their survival. So they're under a lot of pressure, and they're extremely important, so we need to try and protect them. So to bring that to a more local level, in the Caloosahatchee, historically there's been really dense beds of like tape grass and seagrass, kind of just covering the bottom of the Caloosahatchee. For example, a couple of decades ago, up in the upper estuary, there used to be about 1,000 acres of tape grass coverage, and but before 2012, there was a few years where during the dry season, they weren't allowing a lot of fresh water to come down the river, and it led to the salinities reaching high levels in the upper estuary. And in that part of the river, the dominant seagrass species is tape grass, which is a freshwater seagrass. And it has a very low salt tolerance. So when these salinities were allowed to get to these high levels, it led to a massive reduction in the distribution and density of the tape grass. But then after 2012, the Southwest Florida Water Management District and the U.S. Army Corps of Engineers started following the minimum flow regulations, which were established to try and maintain the salinities in the upper estuary low enough to allow for the survival of tape grass. So since 2012, the salinities in that area have been kept low enough to allow for the survival of tape grass. So in terms of trying to restore those dense patches and meadows of tape grass, the salinities are now, like I said, at the correct level, but we still aren't seeing the really dense vegetation that we were seeing before. And one thing that could be keeping the seagrass from really taking off is grazing pressure from manatees and turtles. So it seems like the plants really need some sort of protection from that grazing pressure to be able to fully reestablish and reach the densities that it was at previously. And studies have been done before where they've used exclosures, but generally the exclosures have been small, usually about 1 to 3 square meters. But in this project, we decided to construct much larger exclosures, so they range in area from about 0.1 to 0.3 acres, so much larger than previous studies. And we're hoping that by constructing these big exclosures, we'll be able to reach high densities of seagrass over a larger area in the hopes that once the exclosures come down, we'll have established a large enough density over a large enough area that it will be able to support the grazing pressure once they're exposed to it. So basically the 3 main questions that I was looking at through my study was, 1, are these large exposures effective in allowing for seagrass reestablishment? 2, what areas of the river is the most successful for seagrass growth? And 3, what habitat conditions could be affecting the different success rates between the sites? So now I'll just kind of run through the methods of what we were doing. So obviously the first step was to construct all of our exclosures. We had 6 sites that was along about 10 kilometers or so of the riverbank and extending from Rosen Park, which is a little below the Fort Myers Bridge, up to Beautiful Island. And so our first step was cutting all of these really long pieces of mesh and then taking them out by boat out to these sites where we set up PVC poles around the perimeter of our site, and we connected the mesh to the PVC poles so that it was about 1 meter tall, and that left about 1/4 of a meter or so that we could lay flat along the bottom, hoping that that would deter things from trying to burrow underneath. And all of the exclosures were set up between January 13th and January 25th. So once we had all of the exclosures set up, then we moved on to the planting phase. So we had grown a bunch of seagrass in these little shoe boxes in tanks, and once the shoe boxes were full, we would take out the sods and transport them to the river, where we broke them up into clumps of about 10 shoots, and we planted them in this grid pattern, so it was in rows along the exclosure with 1 clump of 10 every 3 meters. And so we planted tape grass at most of the sites, the freshwater species, except for at Rosen Park, which was the site that was the furthest downriver. And at that site we planted Rupia maritima, which is a more salt-tolerant species, because since it's our farthest downstream site during lower flow in the river, the salinity in that area can reach levels that are too high for the survival of the tape grass, so we opted for this more salt-tolerant species. So once we had all of the sites planted, we began our monthly monitoring. So every month we went out to all of the sites and we measured the densities of the plants, both inside and outside of the exclosures. So we did that using 1 square meter quadrats, and we would count all of the shoots inside of the quadrat. And we did at least 10 of those both inside and at least 10 outside of the exclosures. And we would also note whether there was evidence of grazing, so trying to see if there were maybe small things like snails that were eating the grass or if there were animals that were getting through our exclosure and getting access to our plants. And we would also obviously check the exclosures every time we went out and make sure that we repaired any holes or damage that had occurred. And then we also measured the water quality parameters every time we went out. So that included salinity and turbidity. And we would also measure the light availability at all of the sites using a LICOR. Because light is obviously a very important component of seagrass growth. And we also took sediment samples at every site. So we took small samples sampling the top layer of the sediment at every site. So, and we did that for about 4 months. So now I'll just go over the results that we've been seeing so far. So to answer our first question of whether the large exclosures are effective, I first wanted to see whether we really were seeing an increase in grass densities inside of the exclosures and outside. So in these graphs, the one on the left is inside of the exclosures, the one on the right is outside of the exclosures. And you have the sites along the x-axis and the shoot densities along the y-axis. And so the gray bars are the densities at the end of the study, and then the black bars that you basically can't see are the densities at the beginning of the study. So it's pretty clear that we had a pretty big difference, at least inside of the exclosures, between the beginning and the end of the study. And that was confirmed when I ran some Man-Whitney U tests, and they all came out significant. So we did see a significant increase in the plant densities inside of the exclosures over the study period. And for outside of the exclosures, the scale on the y-axis is the same for both of those graphs, so you can kind of see just how different the outside was compared to the inside. We did have to, at Palm Point, which is one of our kind of centrally located sites, and Beautiful Island, which was our furthest upstream site, we did see a significant increase in the plant densities outside of the exclosures as well. But again, the magnitude of that change was a lot less than inside of the exclosures. So we're definitely seeing, we definitely saw a major increase in the amount of grass throughout the study period. Yes?
Outside the exclosure was not planted?
We planted some just to see whether, so the grass that we grew was nice and tall, and so we wanted to see how quickly they would get eaten if we planted them outside. So we planted some outside, but they usually didn't last very long, and if they did, they were eaten immediately.
So in terms of the relationship between the two, the plants outside were naturally occurring, basically?
Essentially, yeah. So then the other part to answering that question is actually comparing directly the densities inside versus outside of the exclosure. So again, the sites are along the x-axis, densities on the y-axis. The black bars are the average inside densities, and the gray bars are the average outside densities. So again, pretty clear that there's a pretty big difference between inside versus outside of the exclosures, and they all came up significant when I ran them through statistically. So it's pretty easy to conclude that these exclosures were quite successful in allowing for increasing density of the seagrass. So that was exciting. You can kind of see in this picture, but all of this is seagrass that had reached all the way up to the surface. So that was pretty cool. So for answering our second question of which site was the most successful, this gives a kind of a good picture of just the growth that we were seeing at all of the sites. So each graph is a different site, and then along the x-axis is each month, and then the densities are along the y-axis. So at every site, you're seeing this kind of exponential increase in the densities inside of the exclosures, which is great, and that's what we we're hoping to see, because the seagrass, it reproduces through rhizomes, so each plant can send off a bunch. So as it establishes, you expect to see this kind of exponential takeoff. So that was great to see. Rosen Park, which was our furthest downstream site, reached the highest density of an average of 2,830 shoots per square meter, which is pretty crazy. But if you remember, that is where we planted the repia instead of the tape grass, and that species can just also reach a much higher density than the tape grass, just because of its structure. It's got much smaller leaves, and the shoots can grow much closer together. But even just looking at just the sites that we planted tape grass, most of the sites, they were reaching densities between 700 and 1,000 shoots per square meter, except for Beautiful Island. Beautiful Island only reached about 450, but that site we had a hard time keeping grazers out of. They seem to really like that area. So that was a kind of a constant battle. But even with that, we still did see this exponential growth, and it was also good to know that even with only kind of partial removal of the grazing pressure, we were still able to get this growth of seagrass at that site. And so to answer this question of which one was the most successful, I compared the ending densities between all of the sites, and it actually turned out that even though it seems like the averages were different, statistically there was no difference, and that could be, I mean, just from quadrat to quadrat, there could be a fair bit of variance within the sites. So we can't really say which site is really doing the best or is the most conducive to the best seagrass growth. But despite that, it's still interesting to look at some of the other habitat conditions and see what is different between the sites that we chose, because there are other aspects of the plants that could be looked into in the future that could show some more differences between the success at the different sites. So looking at the sediment samples that we collected, I took each sample and I used a sequence of sieves and separated them out into particle size so that I could establish kind of a percent by weight of each particle size. And kind of the most important when thinking about seagrass is how much of the sediment is silt, so the finest particles. There was a study done in Southeast Asia that suggested that in sediments where there's more than a 15% composition of silt, it resulted in a decrease in biomass of the seagrass. So it could be if there's high silt contents in the sediment that could be hindering the seagrass re-establishment here. So when I looked at the silt contents composition at all of our sites, the highest was Marsh Point East, which was the second furthest upstream site. It had an average of a little over 3% silt, but we didn't have tons of samples, and it was highly variable at that site. It ranged from like 0.5% to over 7%. So, but it's still interesting that even the highest percentage that we got was still much less than that value that was found in that other study that said it would have to be over 15% to really be affecting the growth of the seagrass. So it's an interesting aspect to kind of take into consideration, but we would need to do kind of a lot more samples to really be able to tease out any differences between the sites. And then looking at the light, which is obviously a very important component of habitat for seagrass, we looked at how the light availability was changing along the river. So in this graph, the x-axis is the distance from Franklin Lock, so moving downstream as you go this way. And then the y-axis is the 25% depth level, or light depth, sorry. And basically that means it's the depth where there's only 25% of the surface light still available. And that's kind of an important cutoff for seagrass, because most species of seagrass, that's kind of their minimum amount of light that they can have and still be productive. Below that, their photosynthetic rates start not being able to keep up with their respiration rates, and so they start to lose biomass. So looking at that, you can see that as you move downstream, the 25% light depth is getting deeper. So that means that downstream, the light is penetrating farther into the water column. So that means that downstream, the plants can be colonizing deeper into the river, and there's probably more area that is viable habitat for seagrass to colonize. And just as a kind of a visual representation of what we were seeing in terms of the light availability. So in this graph, this is showing the 25% light depths along the river. So the blue colors that's where the 25% light depth is the shallowest. So that's where it's not reaching very far into the river. And then the red colors are where it's the deepest. So that's where the most light is available. So you can kind of see that most of the blues and greens are more in the upstream areas, while the reds and oranges are mostly in the downstream areas, other than that one little patch up there, which could just be slightly more protected from the wind or wave action or things like that. And an interesting kind of water quality parameter that can be used to look at why the light availability is changing along the river is turbidity. So when I looked at the correlation between turbidity and the 25% light depth, it was a very strong negative correlation. And turbidity is basically, it's a measure of the particle load in the water. So if there's a lot of particles in the water, it correlates to a high turbidity. And the reason that could be causing these lower light depths is with more particles, it's obviously blocking more light, absorbing more light, and just preventing the light from getting any deeper. So, and that's a good kind of connection to be making in the Caloosahatchee, because we've also seen that with the high water flows that results in higher turbidities in the river, just from bringing in a lot more particles, and it also causes faster currents in the river, which results in more of the sediment from the bottom being resuspended. So with these high flows, we can be causing higher turbidities, which can be further limiting the light available to the seagrasses. So just to kind of conclude the findings that we've seen so far, basically from the study, I think it's fairly safe to say that the protection from the grazing was a really important part of reestablishing these dense beds of seagrass, and that the large exclosures were definitely very successful in doing that. And it was especially good to see because even within our exclosures, we were getting these large patches of dense grass, and we weren't seeing lots of expansion beyond the fences. So having that large area exclosed allowed for the growth of these large patches that you probably wouldn't be able to get even with a bunch of small exclosures, just because they have a hard time expanding beyond the fencing. We also saw that all of the sites showed large increases in the density of the seagrass, so it's good to see that the habitat is viable in all of these areas for the growth of seagrass. And we also saw that the sediment composition in the areas that we studied doesn't seem to be hindering the growth of the seagrass, so future efforts in those areas should hopefully be successful. And we saw that the light availability does increase moving downstream, but even though that didn't impact the plant densities, it doesn't seem like, so we didn't see any differences in the plant densities between any of our sites. It could be impacting other aspects of the plant growth, which leads me into some further areas that we could look at is looking into the differences in plant biomass between the sites, because even though we weren't seeing differences in the plant densities, the plant biomass could have been different between the different sites, as the plants may not have been, well, as just as dense, may not have been as tall at these different sites, and it could kind of elucidate some of the differences that are actually occurring between the sites. It'll also be interesting to see how the grass fares once the exclosures are taken down, and to see whether we actually succeeded in reaching densities that will be able to survive the grazing pressure or not. And then it'll also be interesting to see what happens when the flow in the river changes, because throughout this whole study they've been very high. So it'll be interesting to see what happens when they do finally go back down. So just to wrap up, I just want to thank SCCF and all of you guys for giving me the opportunity to work on this project and work here. It's been a really wonderful experience. And thank you especially to Rick, who's been my devoted mentor through these past 6 months. So thank you.
Could you touch on, so the enclosures work, it seems like, okay, let's have enclosures, but I'm sure there's permitting and other issues. Could you touch on that a little bit?
Well, I guess I wasn't here for a lot of the permitting aspect of it, but I know that we struggled a bit getting, once we finally got the permit. It took a while, but hopefully now that we've been able to like show that they do work, the permitting process will be a bit easier. But I know that at least in the river they have some concerns with the other animals that are in the river getting caught in the mesh and things like that. But we haven't seen anything get caught, and we've designed it in a way that if anything does manage to get inside of the exclosures, they can find their way back out. But I don't know if Ricky want to add
How long
anything.
were they up officially?
Oh, right, so that because of this sawfish concerns, and National Fisheries Service had concerns about the season of pupping and things like that, so we're limiting the exposure to 3 months of being out there is what they decided, and then we take them out, leave the posts in, and reinstall them next year, and we make them a bit bigger next year. But so what helped with some of these sites was having some existing hay grass. Hay grass doesn't get grazed all the way down by the land, so when you expose it, you don't have to transplant and then come back at LRD early next year. So that helps having some help in between the exposures.
So your permit covers your work for next year, or you have to apply?
Well, we have a permit for this 1 year, and then they said just to send in the same permit, which I did was, and then say we want it for 10 years. So if we could do it, I'll redo it for.
Yeah.
Just on biomass, to measure the biomass, do you have to actually destroy part of the plant, or can you just do size of the blade and get it that way?
So the way that I've tried it was you take one leaf, so you don't destroy the entire shoot, you just take one leaf, and then you measure the length and the various widths, or you can use like scanning. There's like a computer program that you scan it in and it can calculate the area of it, which sounds cool. But obviously you have to do more than just one leaf to, be able to really see the biomass. So you would have to definitely take a fair number of them, but you wouldn't be completely destroying the plant.